Tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you very much for the opportunity to present. And it's always weird when you have to write one of those blobs about yourself. It's a bit like the eulogy we just heard. They're great words, and I'll try to live up to them. ESR, some of you might have heard a lot about it in recent days, but some of you might not be familiar with what are the kind of things that we get up to. We're trying to keep people and communities safe and healthy and prosperous through actually having great science. And our new CEO, who's only recently joined us, very succinctly summarized that by that we detect, we then connect the dots, and then protect. Which means that we're not only doing this for infectious diseases, but also in forensics, in the environment, and across a whole different suite of things where you might not necessarily see the science, but it's supporting it. So if you get pulled over for drug roadside testing, that's us. If you hear about microplastics in the water, that's us as well. So today we will of course be focusing on public health, but a bit about forensics as well, because this pandemic has meant that we have had to become genomic detectives. And to talk a tiny bit about genomics and where it's come from, this is what some of you might know as the Human Genome Project, which started fairly shortly after I was born, 4 years to be exact, shows my age, but it was a 15-year project on trying to disentangle and read the human genome. And that is how long it took all those researchers to come up with technology so that we can now understand human disease. And as you just heard, we actually have been implementing some of that in diagnostic settings, where now within a couple of days, they can read a human genome. And that's because the technology has evolved very rapidly. So some of you might be familiar with Moore's law, which says that computers get twice as fast every year. That's that white line there on the graph. The green line is how much cheaper sequencing has become. So on the y-axis, you have the cost of sequencing a complete human genome. The human genome is thousands of times larger than a viral genome. And you can see that there at the right-hand side, we've reached what they call the $1,000 genome. So that has come down very rapidly, and we can actually now more cheaply sequence something than to store it. Which has also meant that there's been great advancements in technology. Computers, when they first arrived, there were these big machines in factory halls that physical bugs were appearing in and causing problems. The same thing was for sequencers. There were these large, bulky machines that had to sit in a very bespoke laboratory and had to be monitored quite quickly. That left-hand picture is actually someone sequencing the DNA of a snake in the jungle. That is now a reality. That is how far we have come. That also meant they could take that technology to Africa to, in real time, study the Ebola and Zika virus in both South America and Africa. And what you see there is an overview of the sort of the family tree of the virus for Ebola they found that has been colored according to those mutations that occur in that virus. And that's also then projected on the map. So it tells you something about where it started and how it spread over the country. So in the world of sequencing and infectious disease in particular, we're now looking at what we call a genomics-informed, real-time, hopefully at some point global, pathogen surveillance system. And all these things factor in, and this pandemic has brought all of that to the fore, from sample collection to sample analysis, and then to reporting. And what you see now, where this genomics reporting is informing our government, is something that we've been fighting for and striving for to make happen. So this is the first pandemic in what I would call the genomics era, where these types of technologies are widespread and are being used in a day-to-day basis. So on the 31st of December, the World Health Organization was notified of this new emerging virus. On the 12th of January, we had the genome sequence of this virus. If you compare that to its most recent relative, the other SARS virus, That took more than a year. We're talking weeks. That meant that we could start developing protocols and that we could get those diagnostic assays ready, that everyone know that gets one of those swabs, those tests could be designed based on that genomic sequence. On the 23rd of January, the first sequencing protocol to extract this sequence of this virus in a routine fashion had been published. That is an unprecedented speed. Of course, we also wanted to get New Zealand or ESR ready. Seems to be an echo.
That's all right, I'm just trying to
Okay.
this on my phone.
So we touched base with our international colleagues, and on January 28th, a shipment was on its way from the UK to us with the reagents so that we could do sequencing of those genomes as well. Luckily, because this is a global pandemic that was clear by then, everything was shared openly and freely so that everyone could get ready. And we actually got that sequencer, that's the size of it these days, ready to go. First case came to New Zealand on the 1st of March in 2020. It was indicated that they had traveled from the Middle East, and that's where they were infected. Their sample was received by ESR on the 3rd of March, and the genome was running on the 4th of March. And we had it globally available on the 6th of March. So we thought that was doing pretty well, but that's just one genome, right? That's not how you're going to solve a pandemic. So why this genomic surveillance? Just to touch back on that. We want to assist in outbreak investigations. It is an additional line of evidence. One of the things that I like to say is that a virus genome sometimes has a better memory than people because it doesn't forget as easily. It can help to estimate the number of introductions that we're getting. When our borders were open, you know that the virus is coming in, but how many times did it come in? How big was that problem? You can use it to track how transmissible it is, so how fast is it spreading? And also, how is it spreading? So that's these transmission chains that you keep hearing about. So how does that work? And I'll keep it very brief. But we have this genome sequence of the virus that originated likely in the China region, where it jumped from an animal into humans. And what we can do is we can read the genome, and then there's every time that it copies, the virus can make a mistake, and that leaves a mistake in the genome sequence. And if you then infect another person, that mistake is retained. So we can use those mistakes that happen when the virus copies itself to reconstruct a tree, because if a certain mutation is seen in a person and then in a subsequent person, they must have had a link, because people before that did not have that mutation. So these are those trees that we then reconstruct. And we can then translate that to, this is the original sequence, so that would be all A's, that gave it to this person, that then mutated to a C, that then infected these two people. This person then mutated to a T and then infected that person. And this one didn't mutate, but in this next person that mutated into a G. That doesn't happen every time the virus passes in this particular virus, but it provides us with a way that we can track it. So we got to prepare for high volume, and this is what was mentioned in the introduction as well, that we invested early in the technologies to not be able to just do one genome, but to do 200 genomes in 1 week. So we brought in some robots and some slightly bigger sequences. It's still about the size of a printer, so not too bulky. But of course, during all of that, the country also went into lockdown. Systems were under pressure. A lot of us were being at home while trying to work, with kids being home from school. So it was not always easy going, but I think we got there. So we've been sequencing this virus from the very first case that made it into New Zealand. And in the first wave, we have sequenced nearly 650 genomes from the about 1,200 cases that were in that first wave. Most of the areas were well represented, and we could provide links for those people for which they had no idea how or where they might have gotten infected. And that, of course, helped us understand where measures needed to be put in place and how the virus was behaving within New Zealand. And we were also able to add cases to known clusters based on the genomics. And that's what you can certainly see in the current resurgence. Whenever the ministry has a person that they identify with the virus, but they don't know where or how they might have picked it up. With genomics, we can provide that link. Another thing that we looked at was how effective was the lockdown, because of course this was quite a severe lockdown. We went early, we went hard, and how did that work? And what we can see is that it worked really well, because of all the introductions that we had, only 19% then went on to infect another individual. That is a very low number. So that lockdown really worked, because we had over 227 unique introductions of this virus into New Zealand during that very brief window that our borders were still open. And actually, most of the cases that did lead to onward transmission were from North America and Europe. And that is to do with the fact that our borders were close to Asia Pacific, but they weren't close to Europe and America just yet. So there's a lot of cases came in, and they were not managed, and that's where there was subsequent spread. And then the thing that is keeping us up these days, the resurgence. The virus came back to New Zealand. The first case we had sequenced within 24 hours of it being detected. And this is the thing that you also hear on the news. We all of a sudden, we know about genomic lineages. This says something about the mutations that have happened previously. And this was the particular B111. It had not been seen in New Zealand since March, which meant that it was very unlikely that it was one of these slow burning embers, because 3 months of that virus sitting there, that's unlikely. And the other thing that we did also look at was linked to one of the known managed isolation cases. And for this particular outbreak, it was not. And then we had one case that was actually linked to a managed isolation facility. Now, all of these things, I think, are great. They're helping us prepare. They're helping us make these decisions based on data. But of course, we like to think about the future as well. And we're preparing for the next step. One of the things that we're trying to do is to move genomics to the bedside, because in some international labs, they've been able to do some of these things at the hospital, which means that much quicker results. But in certain bacteria can also mean quicker treatment. So we've done some proof of principle studies on very urgent cases with sepsis, where within 2 hours of sequencing right at the bedside, of course, we simulated that in the lab, but within 2 hours, we knew which drug we should be able to give to that person, because sometimes they can be drug-resistant bacteria. So we knew exactly which drugs we shouldn't give, which then also means we know the one we should give. And we knew exactly what species was present within that. And I think that if we're talking about a better healthcare system in New Zealand for these infectious diseases, these technologies are there in the time that they're quick enough, they're good enough that we can start thinking about changing the way we use those. So we actually made this little setup. That's a tiny little computer. And some of the people that have kids or grandkids that love gaming, you might hear about these gaming cards that are insanely fast. This is a miniature version about it. It's about that big. Costs you $200, attached to a sequencer, a power bank, and some storage that can do the job. It doesn't have to be very expensive anymore. We can start thinking about this. And the other thing that we're working on as well is that we know that the swab into the nose or throat is not very comfortable. And if we want to know how this virus is tracking, we need to get as many people to get tested if they show symptoms or if they're asked to. We don't live in a country where we're going to force people to do that. So we want to make it as comfortable for them as possible. And we also want to be able to do more tests, because if we're going into this mode where there might be some community transmission, we want to be keeping the finger on the pulse at the border, in those communities. So we need higher volume testing, quicker testing, more often testing. And that's also a piece of work that we're wanted to take on and that we're looking into to try to help prepare for the next step. I just want to acknowledge, and that was mentioned as well, is that This is a true team effort, not just within ESR and the ministries, but also with our wider scientific community. People from all kinds of fields have been pulled into this response to bring the right minds together, to make these interpretations, to set up these technologies, to work in a way where they're actually informing our decision making. And we consider our genomics team to be a proud member of the team of 5 million. This is made possible by a research grant from ENVI and funding through the Ministry of Health for our surveillance work. And I'm happy to take any questions.
That's what we're looking forward for you. Yes. And that,